In the last class we had seen how the new characters are being formed and how the chromosomal theory, how it was being given by Sutton and Boveri based on Mendelian principle. Similarly, as Sutton and Boveri, he was a student in the Columbia University, he gave the chromosomal theory in 1902. There Mendel was also there. He coined the term, a term as crossing over. He gave the term as crossing over. Now, what is crossing over? Crossing over, if we see, it's a biological process, and in this process we know that the chromatids, the non-sister chromatids are being exchanged, and this crossing over is a biological process and by which new recombinants are being formed. It takes place in 3 steps. Crossing over takes place in 3 steps. First, synapsis takes place, then tetrad formation, crossing over of non-sister chromatids, and fourth is terminalization. So, crossing over, it takes place in 4 steps. Synapsis. What is synapsis? Synapsis, basically it means pairing. If we go back, crossing over, it will always takes place in meiosis, that is germinal. It is also known as, it takes place in the germ cells, that is why it is also known as the germinal meiosis, meiotic development. So, if we see in the diagram, suppose these are the chromatids, it is a bivalent, 2 bivalents, when they are joined by a centromere, now it will be known as a tetrad. Now this is a tetrad. If we separate it, then it will be known as a bivalent. Similarly, one more tetrad, 2 bivalents or 2 synapses, pairing will take place. First pairing will take place. Okay, these will come and lie side by side. Then again this tetrad, it will come and it will lie together. This kind of tetrads will be formed. Now this is one tetrad. This is another tetrad. Synapsis means basically they will come and lie side by side. Means these two will come and lie side by side, the 2 chromatids. And then in the second part, tetrad formation, it will 2 bivalent. These 2 are known as bivalents. This is one bivalent. This is one bivalent. So now this will be known as a tetrad. The 2 bivalents, when it will be joined together with the help of a centromere, now it is a tetrad. So in tetrad formation, what is happening? The tetrad is formed. 2 tetrads, these 2 also will come and lie side by side. This happens in the zygotine stage. This happens in the zygotine stage of prophase 1. This is happening in prophase 1 of meiosis. Meiosis 1, prophase 1. We know that it is divided into 5 substages, of which in the zygotine stage tetrad will be formed. Next, in the pachytene stage, what happens? Prophase 1 again, in the pachytene stage, crossing over will take place between, this is one sister chromatid, this is one sister chromatid, these 2 are sister chromatids, this is one homologous chromosome, this is one homologous chromosomes. So crossing over takes place between non-homologous chromosomes, means this pair and this, if crossing over is taking place, any part crossing over will take place. Suppose this part crossing over has taken place, and suppose this part crossing over has occurred. So crossing over has taken place. Now in terminalization, these will separate with the crossed over parts. This will separate with the crossed over parts, means this way crossed over parts. This is the crossed over part. So terminalization means it will separate with the crossed over parts. Now if we see, if we are separating it again, when it is recombinants will be formed, one part, then this is the second one, then this is the third one, and this is the fourth one. Tetrad, if you are seeing, two are recombinant form with the crossed over parts. Two chromatids are with the crossed over parts and two are the parental forms. So this is what is happening in crossover. It takes place in 4 steps: synapsis, tetrad formation, crossing over of non-sister chromatids, and terminalization. This is how it will take place. It all happens in the meiosis 1, prophase 1, all the crossing over will take place. This crossing over, which was told by Mendel in 1910, this same thing was being told by Sutton and Boveri in 1903. What did Sutton say? Sutton had formed a comparison. Sutton compared Mendelian's laws and the chromosomes, and then he told that whatever is happening, how the chromosome is dividing, similarly the Mendel's factors, they were also dividing in the same way. It was being told by Sutton in 1903. See, we can say this chromosomal theory of inheritance and crossing over also, it was given so nicely by Mendel. In the monohybrid cross, if you see, there was no variations in the monohybrid cross of Mendel. There was no variation. That was being told by Sutton, that when Mendel gave the monohybrid cross, there was no variation, because it could be simply because the genes were located very far from the chromosomes. That's why they failed. That's why no recombination would form. But even in the dihybrid cross, Mendel's dihybrid ratio maintained is 9:3:1. In this also he was unable to give about anything about recombination. It could have happened because in the homologous chromosomes, what the Mendel had been done, what Mendel was doing about the P, in that he gave the 7 characters. In the 7 characters, when he went for the dihybrid cross also, it could be so that the genes, they were not located very far, they were located very close, and Mendel failed to mention the linked genes. He failed to mention the linked genes, because as and when the genes were following, and they were not following the typical Mendelian ratio of the monohybrid cross, that is 3:1. It was deviating, means it was going for 9:3:3:1 dihybrid ratio. According to Mendel, the dominant trait should show itself, but somewhere it was deviating. So Mendel failed to explain the linked genes. That was being proved by Sutton in 1903. Along with the dihybrid cross, he was able to tell that yes, the dihybrid cross, it so happened that the linked genes, they failed to separate. So hence the ratio maintains. In the monohybrid the ratio maintains, and in dihybrid it was not following the Mendelian ratio of 3:1, but it deviated to 9:3:3:1 Mendelian ratio in the dihybrid. Here itself it shows that yes, linked genes were present, but Mendel couldn't notice that. It was Sutton who noticed this, and he gave it in its chromosomal theory of inheritance. The same thing what Morgan also told. Morgan was working on fruit flies. The crossing over what Mendel gave, he gave it in 1910. Morgan, he gave it in 1910. Sutton gave the theory, chromosomal theory of inheritance. Sutton gave chromosomal theory of inheritance in 1902. In 1903, Sutton was able to form the comparison. He was able to compare between the linked genes and the Mendelian ratios, monohybrid and dihybrid ratio. He was able to comprehend, because that time Mendel had given the ratio, but that time Mendel was not clear about the linked genes. So that was being given by, that comparison was given by Sutton. Now in 1910, Morgan also was belonging to the same university, Columbia University, in which in 1903 Sutton was the student, Columbia University, New York. So in fruit flies, what was being observed, that why Mendel took fruit flies, they were also known as Drosophila. The scientific name for fruit fly is Drosophila melanogaster. That means Drosophila melanogaster means black bellied dew lovers, means they used to feed on dew. So that's why fruit flies, the name is Drosophila melanogaster. Why did Mendel choose Drosophila? Why? Because Drosophila, the generation time is 12 to 14 days, means it will arise, then it will reproduce and it will die. Then it can be generated throughout the year. Throughout the year it can be grown in the culture media. Then very few things are required to grow food fry, means a mashed banana or yeast, very few culture media, very few materials needed for culture media. Then fourth, why did he choose Drosophila melanogaster? Because a single mating produces large progeny. Mating produces large progeny. And last, fifth point, and the fifth point, if we see that why it was chosen, because days it will take less time, it can be grown throughout the year, very few materials are needed, single mating produces large progeny, this is there, and the most important is that it has got only 4 chromosomes, only 4 chromosomes, of which 3 are autosomes and the 4th pair, that is the last pair, it is the sex chromosome. Drosophila melanogaster, the sex chromosome, it is forming the XX and XY type in Drosophila melanogaster. It is XX and XY, that is XX belongs to the female, XY is for the male, and which in this the Y, it is not Y actually, in Drosophila this Y is replaced by J, it is J shaped, it is J shaped. In Drosophila melanogaster the male has got J shaped, means basically it will be deciding factor, it is like non-dysfunctional, means it is not functional, the J is not functional in Drosophila melanogaster. These were the points by which Morgan started with the crossing over and linkage. Crossing over I had already explained. Now what is linkage? Linkage, as I already told, which was given by Sutton, linked because they failed to separate. What failed to separate? What is linkage? Linkage means, suppose this is the chromosome, these are the genes which are present, a homologous chromosome. So what happens during crossing over, when crossing over has to take place, suppose this is one and this is other homologous. So when crossing over takes place, what is happened? It has to take place between this segment and this segment, or suppose this segment, but what happens that this full genes, sometimes this full genes, they do not separate because they are lying so close to each other, they do not separate and they are transferred as such. So what is happening, what combination we are getting? We are getting this combination A, B, C, D, E, and here the crossed over segment, here A, B, it comes. So these are known as the linked genes. They are carried all together because only a small segment needs to be present for crossing over to take place. But when crossing over takes place, instead of A and B, the full genes A, B, C, D, E, they undergo crossing over. That means for recombination they are occurring together. So these genes which fail to separate during crossing over, these genes are known as linked genes. These genes are known as linked genes. These genes are known as linked genes. And the process where the linked genes are at the time of crossing over means the linked genes are crossed over as such. So this is known as linkage. The process of linked genes being at the time of crossing over, the linked genes are transferred as such. So that process is known as linkage. And if you see, because the linked genes are carried as such, so there is no recombination because new characters are not formed. Why new characters are not formed? Because in the place where this segment has to be, only this segments need to be crossed over, the complete set comes as though. That means no new characters are being formed. There is no recombination. It is passed over as such. So there is little recombination because where linkage is more, means the linked genes are carried as such, the recombination rates are very less, and when the linked genes are located far apart. Suppose there is quite a distance between them. When there is distance, this is one, this is one, this. So if there is a distance between them, we can see when the crossing over takes place, yes, you get a new combination, AB, AB, a new combination we will get. It is only possible when the genes, the linked genes are present far from each other, means the genes are present far from each other. If they are present at a quite good distance, then when recombination takes place, means when crossing over takes place, a new recombination will be formed. But when the genes are located very close near to the in the chromosomes, they are located very close, there is very little chances of any new recombination to takes place, and hence the recombination rate is very low. So that is inversely proportional. Means if linkage is more, recombination rate is less. If linkage is less, recombination rate is more. That was being given by Morgan. That was being given by Morgan. Now we had seen like how in Morgan had given in 1910 he had given along with his students Sturtevant, Muller and Bridges. These were his students, Morgan students. In 1916 Bridges gave a theory, that is non-disjunction of chromosome. That was given by Bridges. In 1916 he gave the non-disjunction of chromosomes. Non-disjunction of chromosomes means the chromosomes fail to separate. Non-disjunction, basically it is also defining linkage. Hence he was able to prove that yes, chromosomal theory of inheritance, it is based on the linkage. That was again given by the Bridges. In 1902 we know that Sutton gave. In 1905 Bateson, Saunders and Punnett, these three, they worked on Lathyrus odoratus, that is sweet pea. They worked on Lathyrus odoratus. They took two characters, that is purple long and red yellow. They took two characters of sweet pea, purple long and red yellow. What they found, that if it is a dihybrid cross, it should follow Mendelian ratio, that is 9:3:3:1. If it is a dihybrid cross, according to Mendel, the ratio should be 9:3:3:1, but it deviated. We are in the F1, we are getting all heterozygous this way. Then in the, but it will be still dominant. It will be purple long in the heterozygous stage. When it is allowed to self cross, we get this ratio in F2 generation, that is 14:1:1:3, which is far different from what Mendel had given. If Mendel had given, that is 9:3:3:1, it has deviated, which shows that the chromosomes failed to separate. That's why this deviation, that's why this ratio, this was being given by these three scientists, Bateson, Saunders and Punnett. And based on the findings of Sutton in 1903 and in Bateson, Saunders and Punnett, they gave in 1905, based on their theory. In 1916 Bridges was able to give a theory about non-disjunction of chromosomes, how the chromosomes fail to separate because there is so much linkage. Then again we come to the fruit fly. This is how the findings took place, means how they are deviating from the dihybrid cross of Mendel, means why they are deviating, why this ratio is deviating. The ratio is deviating just because the chromosomes fail to separate. If the chromosomes are failing to separate, means what are failing to separate? That means the genes are failing to separate. The genes are carried as such in chromosomes. Why the genes are carried as such in chromosomes? Because these are linked genes. They are not separated during crossing over, and hence it was given as non-disjunction of chromosomes. And the ratios also, if you can see, the ratios are also differing from the normal ratio of a dihybrid cross what Mendel had given, that is the 9:3:3:1. We had seen that in Morgan when he had done, he did with the red-eyed female, red-eyed and the white-eyed male. When he crossed the red-eyed female with the white-eyed female, if in the F1 progeny we are getting a heterozygous red-eyed female and homozygous red-eyed female. Then in the F1 generation we are seeing, we are getting red-eyed male and a white-eyed male. Okay, so it is forming the normal, the Mendelian ratio, that is 3:1, because there are 3 red-eyed and only 1 white-eyed. 3 red-eyed and 1 white-eyed. So it is following the Mendelian ratio. But when the F1 generation is being again crossed, that is, if we are taking the white-eyed female, purely homozygous, if we are taking a white-eyed female, and if we are crossing it with a red-eyed male, then what we are getting? We are getting a heterozygous, a heterozygous red-eyed female, and then we are getting a white-eyed female, then we are getting a red-eyed male, and we are getting a white-eyed male. That it is following the ratio 1:1:1:1. What I want to say here it is that the recombinant rates are very less. We are getting very less recombinations in this. So similarly, when Mendel, it is given in your book also, the diagram is given, figure is given, we had seen that Mendel had crossed the yellow bodied white-eyed with the white-eyed miniature wings, and cross A cross B, it is given in the book. If you see in the yellow bodied white-eyed, we are getting recombination rate as 1.3%. Whereas in the white-eyed miniature wings, we are getting recombination rates as 37.2%. Why there is such a differentiation between 1.3% and C 37.2%? Why? Because the yellow bodied white-eyed, in that the linked genes are present. There is so much linkage, that is why the recombination rate is very low, that is 1.3%. But if in the white-eyed miniature wings, the parental, which was the parental form, in that it had white-eyed miniature wings, the recombination rates are very high. That is 37.2% recombination rates are present. That means genes in this are loosely arranged. In this, genes are loosely arranged, and here genes are tightly packed. Here genes are tightly packed. So the white-eyed miniature wings, why it is loosely arranged, genes are loosely arranged and recombination rates are high? Because it is a sex-linked inheritance, and this white eyes and miniature wings, this is carried in the X. In the sex chromosome, it is carried in the X chromosome. You had seen here also white-eyed, if you see white-eyed, here white-eyed, it is also present in the X, here white-eyed, it is present in the X, here you are seeing white-eyed, it is present in the X, because in Drosophila, Y is non-functional, it is not Y genes. Here we are explaining it as XY just because to differentiate it as a male Drosophila and a female Drosophila, whereas it is no such Y type chromosome is not there, it is a J-shaped chromosome and it is non-functional in males. That's why they are the ones who are determining whether the fruit fly will be a male, the progeny will be male, or the progeny will be female. It is decided by the male Drosophila, and because it is white-eyed, it is found in the sex-linked. It is white eyes miniature wings. This is sex-linked and it is found in the X chromosome. That is being passed. So it only happens, the recombination rates you would have seen in your that will be given in your book. Diagram is given cross A and cross B, in which if in one you will see the recombination rate is 1.3%, and in the other you are seeing the recombination rate is 37.2%. It is only because of the linked genes. In one it is tightly packed, in another it is loosely arranged. As I mentioned earlier, that Morgan had three students who worked with him, Bridges, Muller and Sturtevant, upon which Alfred Sturtevant, the student of Morgan, he gave way to gene maps or the genetic mapping. Gene mapping or genetic mapping, it was given by his student Sturtevant. He gave the gene map or the gene mapping. What is gene mapping? Basically, he studied in some chromosome, in the Drosophila chromosome, because exactly where this gene is present, apart from that, at so much distance, the other gene will be present. Then suppose C, D, E, F, the distance between B and F, that distance is also determined. He did it in Drosophila. Gene mapping, he started off with Drosophila. In the chromosome he had observed. Why? Because only 4 chromosomes are there. That's why it was easy to study the gene on their chromosomes. He found that there is a particular distance is there between the genes, and that distance is maintained. If the same genes are being transferred from one generation to another generation, that distance is being maintained. That is known as gene mapping, and it was being given by Sturtevant. He is the student of Morgan. Based on that, he gave the as centimorgan, cm, meter. Just like meter, it is known as Morgan or decimorgan. In in honor of the Professor Morgan, under whom he had continued his gene mapping. Just to honor him, he had given the distance in honor of him as decimorgan, centimorgan and morgan. That was being given by Sturtevant. Non-disjunction of chromosomes was given by Bridges, as I said, in 1916. He did it, what is non-disjunction basically? Non-disjunction. What is non-disjunction is that when the chromosomes fail to separate at the time of metaphase stage, when these lie in the metaphase stage, we know that it will lie in the equator. At this time, all the chromosomes, it will come and lie at the equator, at the metaphase stage. This stage it will lie at the equator. So when it lies at the equator, what happens? It should separate the centromeres along with the chromatid, it should separate to the opposite poles when it is at the metaphase stage. What happens during this stage? Sometimes it fails to separate, the centromere fails to separate. That means in one unequal it will go, in some it will, in the next stage if we see, suppose two gametes has to be formed, in one complete is going and in the other one more extra set goes. If an extra set goes in the other gamete, the two gametes should have equal, means equal division should be there, but when the equal division is not taking place in the gamete, when one gamete an extra set of chromosome goes, what happens? Why it happens? It is because of non-disjunction of chromosomes. That is, the chromosomes fails to separate from the centromere. When the chromosomes fail to separate during the centromere, a complete set of chromosomes pass to the other gamete. This is what was shown by Bridges in white-eyed female and in the red-eyed male. When he crossed the white-eyed female with the red-eyed male, what they happened? In one they are getting normal eggs here, normal eggs. Okay, we are taking, if the normal eggs are taken and the sperm of this is the normal eggs, and here the white-eyed female, what we are getting? A red-eyed female. This is the X from the male, X chromosome, and this one is the Y or the non-disjunctional J. We are getting a red-eyed female, we are getting a white-eyed female, and here what is happening when non-disjunction goes? That's what I said. In one a normal egg went, here same, and once in another gamete if an extra chromosome goes, goes, what will happen? It should be a single gamete, this two, when it will go, and if one more sperm comes, if it is this one, if another goes, this is an X chromosome of the Drosophila, if goes, it becomes 2 already it is present, and one more it becomes super female, XXX, and hence it will die. Similarly here, null X, if it is going null, nothing is there, and if the sperm goes and it is X, then what we are getting? It is an exceptional red-eyed male, because the character is carried out in the X chromosome. So here exceptional red-eyed male. Here again we are getting WW, then it is exceptional white-eyed. Here suppose this is the Y chromosomes, it is going the sperm of the Drosophila, so it is XXX, that is exceptional female which is white-eyed. And here suppose this will go, this is X, okay. So when it will go, so it is already, this is null and devoid of it, it dies, this will die. So this is non-disjunction of chromosomes, just to make you understand what is linkage and ring combination. Non-disjunction of chromosomes, why? Because the chromosomes fail to separate at the metaphase stage. When they fail to separate, an extra copy will go to the other gamete, and if the extra copy goes to the other gamete and if fertilization takes place, what will be the result? That is being showed in this, and which was being given by Bridges in 1916, non-disjunction of chromosome, and hence he was able to prove the chromosomal theory of inheritance with a very proper thing, that yes, linkage takes place. Why linkage takes place? Because due to non-disjunction of chromosomes, the chromosomes failed to separate. They were able to provide proof that what Sutton and Boveri has said is very true, and this is how it takes place, and this is how chromosomal theory of inheritance will carry on. In the next class, we will deal with Punnett square. Till then, stay safe, stay home. Thank you.